Since 1977, the Saba Lions Club has been a pillar of service and community care on the island. What began with 20 committed members has grown into a legacy of giving back, supporting those in need, and advancing the global mission of Lions Club International. From cultural events to signature service projects like Meals on Wheels and the Food Bank, the Saba Lions Club supports the elderly, disabled, and struggling families with compassion and consistency. Their health programs, like annual eye care support and glucose fairs, continue to promote wellness across the island. The club is also a champion for the environment, proudly maintaining an adopted hiking trail and backing local cleanup efforts to protect Saba's natural beauty. Several of its members have been honored with the prestigious Melvin Jones Fellow Award for their outstanding dedication, proof that the spirit of service is alive and well on Saba. The Saba Lions Club, serving with heart for the good of all.
Is this working now? There we go. Yay! Okay. Okay. Our event is not possible without the help of many sponsors and donors. First and foremost, we'd like to thank the Lions Club. They bring our live streaming to the event. So you guys are here in person, but we are also live streaming this to our Facebook page. And this is possible because of their generous donation. Want me to switch again? Third time's the charm, maybe. Okay. I think you've heard enough about the Lions Club, so I'll move on. We also would like to thank CCG, which is the Caribbean Culture Fund, and public entity Saba for supporting us for 22 years now. And a big thank you to all of our sponsors. Several of them are actually here, so big round of applause for you guys. And our speaker tonight has been graciously accommodated by El Momo Cottages. We do have a raffle going on all month long. I don't know if you saw Lynn or me at the beginning. They are $5 a ticket. The drawing is at our final night on October 27th. You do not have to be present to win. Lots of amazing prizes from jewelry, handcrafted bowls, 1 week on a liveaboard, and hotel stays on Saba. And this year, our raffle has an extra special meaning as we are raising funds to support our scientists who will be coming here January through March in 2026 for round 2 of Critter Quest. So we will be bringing probably about 12 scientists back to the island and do a more in-depth study about mostly insects, but also other invertebrates. And to find out more about this, stay tuned for McKeel's presentation on October 5th, which I just talked about, and that will be at Calibri Cafe, and McKeel is somewhere. There he is. Yeah, so McKeel will be our speaker on Sunday night. It's going to be a great presentation. He's going to give us some preliminary results from the Critter Quest expedition that happened earlier this year. And if you attend any of our field projects throughout the month, you will win a free hat, your choice of either a bucket hat or a baseball hat. And they are both made from recycled materials. And now, our main event, who finally landed here yesterday. What? I don't know the menu, but yeah. So you are invited to stay for dinner here tonight at Liam's Cuisine. They have a dinner menu of $15. I believe there's pork and shrimp tempura. It sounds delicious. So if you're interested in staying for dinner, it's $15. Highly recommend. You can also pre-order if you don't want to wait. But anyways, back to our main presenter. And of course, if you know me, I love puns. So all of my intros have puns. I'm sorry. So get ready to see the future of coral conservation with none other than Joe Oliver. As the Director of Restoration Operations at Coral Vita, Joe leads groundbreaking efforts to bring dying reefs back to life using innovative techniques like microfragmentation and assisted evolution. With over 20 years of experience in marine biology, aquaculture, and underwater operations, Joe's career has taken him from the Curacao Sea Aquarium to Guam's underwater world, and even back to Saba, where he once served as a marine park ranger overseeing the vast Saba Bank. A Wilmington, North Carolina native with a passion for marine life, and born even during a hurricane. No joke. Joe now helps steer Coral Vita's mission to fight climate change beneath the waves, work that earned the organization the prestigious Earthshot Prize. Whether he's growing coral, engaging communities, or cooking up something great on land, Joe brings deep knowledge and infectious energy to everything he does. Let's give a wave of applause for Joe Oliver.
Fantastic. Am I live? Excellent. So my name's Joe Oliver. Nice to meet everybody, those of you that haven't met before. It's good to be back on Saba. I'm here because of See and Learn, so thank you to Lynn, Emily, and all the sponsors for making this possible, convincing me to come back. And I'm excited to talk about several different things related to coral restoration and then coral conservation and the projects going on here in Saba. So without further ado, I'm going to start first with a little bit about Coral Vita and our mission and what we do, and then I'll pause for some Q&A time, and then after that, I'll then go into the more Saba-specific aspects of the project that's been conducted here. So this is just my opening slide, a little bit about Coral Vita. What we are is a company, so we are for-profit, not instead of a non-for-profit or an NGO, based on the idea that reefs in the world's oceans are a multi-trillion-dollar resource. There is so much exploitation, and the amount that is exploited, extracted, versus the amount that's put back into it is incredibly disproportional. And one of the biggest downsides to the excellent work done by NGOs and nonprofit organizations is that the funding pool is limited. The amount of resources are limited. Your timescale often is limited by either grant funding, project timelines, whatever it may be. The thing that Coral Vita does different is that we have tried to tap into bigger and long-term sustainable funding pools and eventually want to be able to hold those who are being a detriment to the ocean accountable and start getting them to pay for the things that they fix, and not just in one place or another, but on a large scale. My goal is to put myself out of job and hopefully get to the point where reefs and understanding, education, and conservation is at the forefront, and then I can go back to fishing. So moving on. So again, our main emphasis is on land-based facilities. So we have our pilot facility, which is in Grand Bahama. It's in Freeport. And we work specifically at this facility with just Caribbean corals only. We also do have a facility in partnership with KAUST, which is a large research facility in Saudi Arabia, in which we run their development nursery, which is currently the largest of its kind in the world. And we're currently in the progress of developing several future projects, not only in the Caribbean, but globally. This is just a subset of the things that we do. What you see here on the left is one of our coral cookies. This is a fusion structure, and what you're seeing is microfragments. So we take large coral colonies, we're able to produce large numbers of smaller fragments in high fidelity that we're able to then monitor and track over time. The reason that we do this is that we're able to harness coral's natural healing abilities, and there's often augmented healing after there's damage or stress. So we cut them up on purpose to then harness this natural healing and growth factor. Same thing with us. If you break a bone, your body starts sending nutrients, healing factors to that area. Same thing with the corals, but we use this to then encourage them to grow quicker and then also to refuse back together. So what you see in the hands here is that all of these coral microfragments have now starting to grow back together and fuse, which is excellent because those are only about 3 months old in that photo. So the rate that we're able to produce volumes of coral is significantly higher than what has been traditional and standard techniques up until this point. But the other thing that we're able to do by doing this is that, and this particular object is the size that it is for a reason. Corals have the ability to transfer photosynthate or metabolic energy, sugars, healing things within about 10 centimeters. So the idea is that any part of this coral colony can aid, assist, or feed any other part of the coral colony. In the middle photo, you can see some of our staff just working with the microfragments, and then another aspect of our enterprise is we also incorporate spawning, so natural coral reproduction from the wild. We go out and capture wild spawn, settle that, and then use those new genetic individuals to then also augment our grow out and restoration programs. But a big part of what we do is not just growing coral, but is also outreach and education. We serve as a hub for a lot of innovation, a lot of marine science research in the Bahamas. At this point, we have 12 Bahamian staff that run the facility and are now starting to take over my job, which is great. We've got 2 individuals that are now starting to run aspects of our field and our restoration operations, which is where we need to be. This will all work better if the people that are the most at risk and have the most need to gain from the resource are the ones that are then the most involved in maintaining, conserving, and eventually growing that resource. We also partner with a lot of other local stakeholders, the Bahamian government, Department of Fisheries, as well as other conservation-related partners. We currently have a mangrove nursery that houses about 40,000 mangrove propagules annually that also get outplanted every year, along with, not in this photo, is a conch hatchery, which has been up and running now for just about a year and is starting to run their first clutch of juvenile conch. So our aim is to also facilitate other conservation and restoration operations. But we have experienced hardship as well. So all of these images are from Hurricane Dorian, which hit the Abacos and Grand Bahama Island in September of 2019. It devastated our facility. We had 32 raceways. We found about 15 of them 30 miles down the island in the bush, all pretty close together. So we were able to reclaim those, put them back together. But as you can see me leaning out the window of that truck, we're fording through 2 and 3 foot deep water. Most of the island was underwater for 24 hours, 10 feet deep. It was extremely bad, but it just gave us a reason to double down on how important what we do is. Because these are some of the most vulnerable communities, and these are the people that benefit the most from having healthy reef ecosystems. Here you can just see some of our broodstock colonies that will then be propagated into those microfragments that we then use for our grow out and restoration operations. So these are all acroporids that you can see currently, and there's mostly palmata, but we work with, right now, about 10 different species. We've worked with up to 22 at one point, which is almost half of the species of Caribbean coral. But we decided to focus more on the ones that are most threatened, that are most endangered, and are most critical for building reef habitats. Another part of what we do is, restoration as a service. That is one of our main funding models. The idea that, again, that reefs are a huge, huge draw between tourism, fisheries, ecosystem exploitation in all different manner, but there's way too little being done to rejuvenate, replenish, and protect these resources. So we are working with developers, coastal property owners, governments, like public entity Saba that brought us here, to really develop these long-term mechanisms. And I know everybody probably sees cruise lines on there, which I don't like either, but they're one of the biggest contributors to damage, and they pay almost nothing in remediation. And at this point, unfortunately, they have billions of dollars, lots of lawyers, and nothing but time. I've actually fought 2 losing battles to cruise port development. So it's way better, if they're gonna get their way, to at least make them on the front end start paying for remediation, paying for relocation, paying for restoration to mitigate the damage that they know they're gonna do. But what goes hand in hand with that is then building a world-class ecotourism facility. And we're currently in the throes of rebuilding our Grand Bahama facility to incorporate a lot more tech and a lot more AV or audiovisual kinds of interfaces, and a lot of what will hopefully be a 3D immersive experience from on land. Because unfortunately, not everybody can go scuba diving, wants to go scuba diving. But when you can see how important these organisms are, and you can see how amazing this ecosystem is from the comfort of dry land, your connection to it becomes stronger, and your will and your need to then protect that also increases. Other things that we do, coral sponsorship. So we do adopt a coral for sponsorship, but we also have corporates and individuals that will adopt entire raceways that will fund the growing of coral that will then be used in our restoration projects. We're also working heavily in technological licensing. We're working on developing several pieces of tech that we're hoping that will not only make our lives easier and make us better at what we do at coral restoration, but we'll also be able to parse out, and some will be open source, some we're gonna have to get paid for. But the idea is that we wanna produce a set of tools that we utilize, but we can also share with the restoration community at large to improve everybody's success. A rising tide floats all boats, and that is one of our main objectives. So one of the projects that we've been working on is for the Global Fund for Coral Reefs, in which they have put together funding packages for different islands, different locations, and looking at restoring reefs that have either strong cultural significance, strong tourism or economic impacts, or have been degraded and resulting in shoreline loss, destruction, and erosion. So one of the reefs that we have been working on did not show up in this slide. Also does not show up in this slide. It's on the next slide. There we go. So this is the area that we've been working with. This is called Banana Bay Reef. This whole area is 34.74 hectares, or I think it's 34,000 square meters, or no, 347,000 square, it's up there. But the idea is that the Global Fund for Coral Reefs has facilitated us restoring a significant chunk of this reef. And this is just one facet of this project. This particular area is about 1.3 hectares, or 13,000 square meters, and has been outplanted with a little over 10,000 coral microfragments, or 1,950, I think 8 of those fusion structures that I showed you earlier, which has at this point doubled our previous ability to outplant corals. It's excellent because we've run into a new kind of problem that at first the concern was, can we get the coral from the farm to the seafloor fast enough in a timely, scalable manner that it is not cost or time prohibitive. Turns out we can. We're actually pretty good at it. The problem is now we've got to work on development and making sure that we're producing higher quality corals, producing a higher volume of them, and ensuring that they then go into the appropriate sites. And so a big part of our process is using the foundational science and understanding of reef ecology to then aim our restoration efforts at areas that, one, need it, but two, will also probably be weighted for success. There's a lot in the restoration community that can be said about site selection. Oftentimes, sites are selected by the donor or the funder, and they're not necessarily in an ideal location or somewhere that will really be successful in the long term. We've had to say no to clients before. We've had to tell people that what they want is, they can give us $1 million, but we can't make what they want where they want it. And we don't want to take those kind of contracts. Our goal is for the reef to be successful, for what we produced to be viable and long term, and then to thrive, survive, thrive, and eventually reproduce. So this graph is just showing a breakdown of the species richness, the biodiversity, and this is what we then use to develop our grow out plan and then our outplant plan. So this is one thing that we have done, is try to come up with a way to standardize coral restoration, because one of the biggest problems that you run into, if you go to pretty much any practitioner anywhere in the world and you go, how do I restore this particular chunk of reef? The answer that you usually get is, well, it depends. Because that's probably the most scientifically accurate answer, because there's a whole host of factors depending on where you are in the world. Even in the Caribbean, it varies from island to island. Even in the Bahamas, there's 700 islands. They all operate a little bit differently. There's some parallels that can be drawn from site to site, but one of the things that we've tried to do is, okay, reefs are complicated, islands are complicated. It's not a one-to-one straightforward system. How can we make this so that anyone with a basic amount of husbandry training and understanding of coral biology can then execute outplants in high volume with a really high degree of fidelity or a really good degree of execution? So one of the things that we do is we've come up with a couple of different sets of standard outplant. This allows us to build either reef building, so based off of your more dominant reef building species. These are your big structure builders. These are your big bouldering and mounding corals that then generate a lot of the basement or pavement of the reef. But we also have our wave breaking and massive SOUs. So these are the ones that, depending on the ask, if the client wants a tourism reef, that's one ask. If you want a reef for wave attenuation. And the reason that this matters is that a healthy coral reef can disrupt up to 97% of wave energy. Just by the way that waves work, the reef will then essentially steals the energy from the bottom of the waves and dissipates a lot of that wave energy. It won't prevent the water from coming up, but it will prevent it from knocking your house down, which is really important. So that's one of these formulas that we offer as part of our suite of things that we can do in restoration projects. Another key part of this is monitoring efforts. And this slide's boring because it's all words, but going through and actually seeing what happened. And that's one of the things that our model allows that many operations, unfortunately, are unable to do continuously, is that we want to monitor the corals not just for 1 year or 2 years, 3 years, 5 years, 7 years, 10 years. Ideally, as long as that we're still running, we want to be learning more and more about what we've done, what works, what doesn't work, and how we can modify our solutions and our methods to be more and more successful over time. So the cool part is this one, when it actually works. So these corals are ones that have been grown from microfragments. Each one of these corals here started out about no bigger than about 1.5 inches, 2 inches, so about 3 to 4 centimeters long. Now, these corals are about 3 years old, but they have survived the worst bleaching event we ever saw in the Bahamas, which was in 2023. And then last year was not great either. So they've done very well. We've seen corals decline everywhere. We've seen it in Saba, I've seen it in Guam, I've seen it in Curacao, Bonaire, you name it. So anytime you see success story like this, and should be noted that these corals all came from within about 100 meters of where they're planted right now. So that's one of the things that we do is we try to specifically target our broodstock corals that we use for propagation to go back to where they came from, because those corals were already growing there. They were already doing well and already healthy. This is a palmata. Now this one is interesting because this one's only about a year old. So these microfragments have been planted earlier this year and have grown out, and you can see you see my mouse? No, you can't see my mouse. But if you look at around the edges, you see that light edge, and then at the end of the polyps, that's not from bleaching. That's the growth area. That's where it's growing down, putting down new skeleton, and the coral tissue has not filled with zooxanthellae, the algae that then give coral their food and energy and color up their tissue. So that's all new growth, which is great because I took this picture 2 weeks ago. And the water temperature in the Bahamas, some corals are a little angry, but some of them are doing really well. And so this is what we want to see. This is a coral colony that started out as about 30 microfragment plugs, same thing, all about 1 centimeter to 2 centimeters squared. And 3 years later, they now are about the size of my laptop, maybe a little bit bigger. But the most important part of that, not only is it big, it's a home. Because corals, like I said before, are the ecosystem engineers. They're a critical part of providing the basis for the food chain, but also habitat. And in parity with literally what the coral reef needs is a juvenile diadema now living under this colony of Acropora that we've outplanted. So for me, that's success. Even better if it spawns. That's what I want, but I'll take juvenile urchins. So before I get too far into why I'm here and why most of you have seen me running around for this last year, I'll pause and take any questions about Coral Vita, our operations, our execution methodology, anything that you guys may have, before I start into the Saba leg of this particular endeavor. Yes?
You mentioned that the coral is growing faster than the fastest that you've been able to do. Is it growing faster than in natural condition? And if so, do you think that it will be strong enough to be that on the continent?
Okay, so that's a pretty good question. So it's two parts, but I'll try to tackle both of those. So the first part of growing the corals faster, and I say that because that is the easiest way to get everyone to understand conceptually what we're trying to do. We can't actually make them grow faster. They have a maximum growth rate that no matter how much stuff we were to pump into them, how much light we were to give them, how much food we would give them, there's a max. Human beings only get but so tall for a reason. We only live but so long for a reason. They have limitations to their metabolism. But what we're able to do is using their natural healing, in which they would grow 10 to 20 times faster, like more area or covering more new territory, we're then put them closer together. So what would normally take, 5 to 10 years to grow something the size of my fist, we can then fill that with microfragments, and it will fuse together within a year or two. So it's really we're not making them grow faster, we're reducing the time until they get to be a larger or a more competent colony. And then the second part was, does that lead to a survival or resilience problem? Is that correct? Okay. At this point, no. Because, yes. Well, I'll let you know in 10 years. If you were to look at the number of coral restoration programs globally running longer than 5 years, there's probably about 10, maybe 15. If you want to go 20 years, there's 3. If you want to go 30 years, there's 1. And she now works for someone else. But there was a lady in Thailand that started the first that I know of, like real legit coral restoration program. And that was I think she started in 89 or 91. I can't remember what it is. So unfortunately, our sample size is really small. And this is a problem that's been going on. Reefs have been on decline since the 50s and 60s. We've just now in the last 20 years started realizing that this is something we can do. And a lot of our techniques and things have come out of actually the aquarium hobby. Because, as a marine biologist, the last thing you want to do is just go start breaking and being mean to a coral colony. But if you're an aquarist and you paid $100 for a piece of coral and it's doubled in size, you can then fragment it, sell those fragments. And a lot of those techniques actually kind of got pirated by the science and restoration community from the aquarium and hobbyist community to lead us to this point where now these techniques are pretty much universal with every restoration organization in the world. So there's been a bit of convergent evolution in methodology from different places in the world. People that talk to each other, but also people that don't talk to each other, have narrowed it down to about the same 4 or 5 techniques that tend to yield long-term success. Now, a good point is, though, with the microfragmentation, we're producing clones. So if the original parent lineage is Rambo and is just not going to die, then it's probably going to be fine. But that's why for us, spawning is such an important part of the equation, because that's the reef's natural resiliency mechanism over time. That's where you see the plasticity in population adaptation over 10 years, 50 years, 100 years, 200 years. Unfortunately, I won't be around long enough to tell you how well that works. But everything that we know to this point has kind of led pretty much everyone down the same set of paths. Great question. Thank you. Yes. Yeah.
So is it a particular way that you get your sample? Do you, like on a plant, or a particular place to cut it, or how do you know that this is where I should be cutting your sample?
That's a very good question.
Countries are trying.
Yes. Yeah.
They've been trying to do this and they all die.
Yeah. So the question is, how do we identify what is the right part of the colony, or which part of the colony is going to be most successful? We're also developing and understanding. Part of our tech suite that we're working on is actually an imaging set that allows us to top-down image all of the corals after we microfragment them, and then see their growth rate, see their healing rate, and then also as we stress them by increasing the temperature, altering the pH, we can then see which corals survive and thrive and which ones fail. But if you were to ask what is the best part of a coral colony to fragment for ideal success, I'm going to be really annoying and say it depends. Because it depends on the species of coral you're working with. With a branching species, your fastest growth is typically out of the last inch or two, or so the last 3 or 4 centimeters from the tip. But with a bouldering coral or a massive coral like a brain coral, it's around the margins, because they actually don't grow out protrusively. They only put a little bit of skeleton on as they get larger and larger, but they expand their footprint around the margins. We're actually doing a couple of experiments now looking at, within the same colony, is there a variation between growth rate from the edge, the center, the apex, and those kinds of things. And we're not doing it with just one. We're doing it with 30 of each species, so that the effects that we see are hopefully not an artifact of just that one coral being exceptionally good at the mean things that we do to it. Yes?
Have you done any work with like trying to hybridize and bringing your micro fragments from different groups together to see if you can make something stronger?
Not. Yes. So the question is, have we tried to hybridize the corals? And the answer is kind of. Again, it's very species specific, because for the most part, most Caribbean corals kind of hate each other if they're different individuals. And you end up with a competition, sort of a graft versus host syndrome kind of battle. But one species, Cervicornis, will actually hybridize. You can get differential genetic individuals to fuse, and they will retain, for a period of time, they will retain their individual genetic footprint, but usually one ends up winning versus the other over time. The way that hybrids are naturally produced is actually through spawning. And that's where you then end up with either new species of corals or more resiliency, because you have a parent that has disease resistance, a parent that has heat resistance, and then you get both of those genes, so now you're resistant to heat and disease. A lot of those things are being talked about in the restoration community, but we're due to the risk of potential harm has been one of the things keeping that from being pushed more. So there's not anyone that's taking corals from Australia and the Red Sea and trying to like smash them together and make a super coral, at least not with the physical corals. Now you can do that with the gametes. You can take sperm from one species, eggs from another, and try to get those to fertilize and be viable, but we're just breaking into that technology now. Currently, the closest thing to that is with spawning, because many species, especially like Dendrogyra cylindrica, so the big pillar coral that almost no one has anymore, they're using a combination of spawn from Curacao, Mexico, Honduras, and I want to say the Dominican Republic, and then they all meet. Someone gets on a plane with a bunch of those vials that I had in that slide earlier, and they all meet in Florida. And the Florida Restoration and Aquarium Lab has been in partnership with Secor, who does sexual coral reproduction for restoration as well. So they stick only to spawning, but they are then using that technique and that technology to then essentially take what would be reproductively extinct populations and still allow them to reproduce under the guise that those new offspring, as they grow, will then be doled out to the donor sites to then replenish their populations. Really good question. Yes?
Question about how do you know what of a site is a good site or a bad site?
So that's a slide I didn't include because it's a lot of science, but I go into it a little bit when I talk about Saba, but we do a series of surveys. So we're doing ecological surveys. Depending on where you are in the world, there's two main acronyms that are, one's AGRA, the other is GCRMN. Don't worry about what the letters mean, but the main thing is that there are a regimented set of rubrics for how to assess a reef ecosystem, and it includes the benthic composition, so what's growing on the bottom, algae, sponges, corals, invertebrate composition. Are there urchins, conchs, lobsters, or things that are detrimental, like fireworms, or things like coral predating snails? You look for those kinds of things. Fish biomass and fish biodiversity. How many kinds of different fish are there, and how many different size classes? How many of them are there? Are they big, small? So we use all of these different rubrics to then go through and assess the reef and see what is the population of corals that are there. Ideally, even a little bit historically, what used to be there, and then we look at the new recruitment, so the new juvenile corals that have spawned and settled out, what's now trying to live here, because the reef knows what it's doing way better than we do. We're making a series of educated guesses based off of the best that we can use science. So we tackle it from the approach of, if we want to restore a particular, if someone draws a circle on the map and says, I want to restore this reef, then we would go do all of these assessments just to see if it's even possible. And then our plan would then be built off of what's there, what should be there, and what's doing well, so that we can then harness what's successful and build on that into the future. Great question. You had one?
I did. I was following up on the cloning question. If you, given your success, is there a marginal maybe problem with a monoculture and maybe a...
Oh, absolutely. So that is a huge problem, and that's been a problem in the restoration community, and one of the biggest pushes now is for genetic diversity. There's an argument to be made that there is a minimum population or colony size for a given area, and depending on who you talk to, it can be argued down to as low as 9. That's the given science, so we double that. So if we're working with brain corals, we have at least 18 broodstock. Ideally more. It depends on the ratio of what the actual footprint and what the grow out plan is, and like what's needed. But we always try to go over, and we're always bringing in new broodstocks. We keep track of all of our broodstock, and I'll show you the tool we use for doing that when I talk about the Saba project. But we always keep track of where corals came from, how they do, how their parent colony does over time, so that we can keep track of winners and that we don't propagate anyone that's going to fail. Awesome. I didn't repeat that one, sorry. Anybody else? Questions before I move on to the Saba specific parts? All right, fantastic. Okay, so why I'm here, and why I was here, and a little bit about what we've done in Saba. So here you can see our Director of Restoration Science in the nice neon pink fins. That weird rope that you see just kind of disappearing off into the distance in the surface is actually an underwater GPS. Well, it's a underwater deployable surface GPS unit. So it has a GPS tracker in it, so because GPS doesn't work underwater. How do you know where everything is? This is one of the tools that we use to identify exactly where the targeted coral colonies were, and then ideally where we would want them to go on the recipient end. So Doc is now deploying that device to the surface to then take the GPS points for those specific corals you see being identified right there. So a bunch of boring stuff at first. There's going to be a lot of abbreviations. I'm going to try to not use too many of them. Or else I'm going to go through some of the maps, a lot of our examples and sort of methodology for the development of this process and this project, and then a little bit about how all of the execution went down. So the main thing, this is just the names of the species that we worked with. So the abbreviation you see on the left is what's called their AGRA code. So that's one of those survey mechanisms or survey rubrics. And this is just how you refer to them in shorthand. Each species, its endangered status, its notes. So we focused ideally on the most critically endangered species and the ones that are most critical for building reef and reef building habitat. And here we are in Saba. So this is the target site. So you can see in this top image both the Black Rock's donor site where the new harbor will be built, along with on the right-hand side in the top right corner is then a box around Hole in the Corner and the coral nursery there at the end of the island. So the goal was to take essentially, we were contracted to remove 250 corals from this blue polygon and this red polygon, and we were told they need to go into that yellow one. So we developed a plan for how we would make sure that we get enough coral of the right species breakdown, make sure we have enough different size classes and biodiversity to ensure an even slice and give all of the different species a pretty even shot across the.
So when they gave you this assignment, you didn't decide where they go?
Yes. So when we took on the project, the recipient site had been designated as part of the original proposal. And so we were subcontracted by Ballast Needham, who is the contractor to build the harbor, to relocate the corals. And that was the pre-established recipient site. That was the box we were told we could play in. And if you see on the bottom right-hand side, that yellow box, there's a red box and a purple box in there. Those were the areas out of that site that we determined that had the most area, the right species composition, and the right physical factors to match the donor site. So there's other parts of that box that we did not use because they were less ideal or not right for the targeted species.
Didn't you say that you normally make that assessment before you take the project on?
Absolutely. That one came baked into the project. But also, the person that wrote the proposal for that project consulted me when they wrote the proposal. So there's a rhyme and a reason to it a little bit.
Was that done by satellite?
Say what?
Was that done by satellite?
So this is satellite image, and then in a bit, some of the other maps have drone imagery, and I'll go over a little bit of that as well. And then, so this is a map generated using those GPS points so it shows all of the different corals. This is essentially where all of the inventory corals ended up. So the different color markers indicate if they which one of the recipient plots, which one of into the nursery, or then the yellow ones, which are on the eastern end of that map, were outliers. They were outside the bounding box that was designated as the high-risk zone for the project. But this is how we actually kept track of all of that information. This is a shot from our proprietary app that we use with Coral Vita. We developed this for managing our inventory and our restoration programs in the Bahamas and other facilities. But we realized that this would be an excellent tool to apply to this particular project to allow us to inventory, track, identify, and then provide the necessary data that also goes along with describing each coral, where it came from, for the decision-making process of where it should eventually go. So we've used this. Each one of those dots, again, represents either an individual coral or a cluster of corals. You can see there's an orange one. Well, you can't see it very well, but there's an orange one highlighted in the middle, and that is that palmata that is in the top right corner. So each coral got a unique identifier that allows us to keep track of it. And now you can see how we actually do some of the more in-depth analysis. So this image is what's called a photomosaic. It's a consolidated image stitched together from about 1,800 photos taken by me swimming very, very carefully over the reef. And then we use a software program to then stitch all of these images together to give us one single high-resolution image. So from this, we can then actually go through and identify individual corals. We can identify their tags and then actually track as we move corals from before and then after. So in the previous slide, if you look in that middle of the big rock in the middle, you can see there's a very large palmata colony there that in that second slide is no longer there. And because we moved pretty much all of him. He's doing well, by the way. I saw him today. That's Apal Orange 18, which is one of my favorites. I tell people don't pick favorites, but it happens sometimes. This is another tool that we use. This is a depth elevation model generated from the same image. And what we did is, as we were doing our assessments and our preliminary surveys, we also deployed a lot of light loggers and temperature loggers at different depths through different parts of the project, the scope of work area. And we use that in combination with the metadata to then sort of develop a depth elevation and light curve model. So to ensure that the corals get where they come from and where they end up matches as closely as possible. But it's also really good for seeing that change over time as well. You can see that large palmata, nice and red, pretty much absent. But the even better part is this, where you can see the whole thing gone. But not only is the entire palmata colony relocated, on the left-hand image is the before, you see all those little orange tags. Those all identify coral colonies that need to be relocated. Notice in the right-hand slide that there are none of those. We managed to clear pretty much everything off of that rock around that colony, as well as all of the main branches. The reason that we did not extract the trunk is because it can be very difficult. And it was actually advised by Eric Meesters to focus on moving more arms than moving the trunks, because the trunks traditionally don't have a great survival rate anyway, whereas the arms do significantly better, because they're also the part of the colony that is most actively growing. Exactly. But again, here's that same slide, one before and then after. And then this next chart then shows our coral relocation moves by the day. So you can see as we went through the project, we started out a little slow, and then we kind of picked up speed as we got better at it. We got a better understanding, and as the team that we were working with from the SCF here really leveled up their ability to execute as well. So I really could not have executed this project without the help from the guys from the Conservation Foundation, and ladies, sorry. Because really it took a large team to get all of this done. We probably could have done more or done it faster if we had thrown more people at it, but that's also more resources, and it becomes significantly more complicated. So given the fairly small team of about 8 that we had on a daily basis, we were able to relocate a significant number of corals. So the target for the project was stipulated in the contract was originally 250, and in total we ended up moving 387 coral colonies. The even better part, though, is that once we left the tools and the know-how for the organizations here on Saba to continue that work, and they've done a phenomenal job since we left. So I think at this point the running total of corals relocated throughout the end of where our responsibility ended is close to 1,000, if not over 1,000 coral colonies. So. And that's a pretty significant effort, because the next slide is going to show you that it's not all easy. Don't worry, she's not about to hit the coral with a hammer. She's actually, if you see next to her tank, she's hammering in the ID tags. So that is an Orbicella, so that's a star coral colony that has been tagged for relocation. So the first part of the project was survey, identify the corals that need to be moved, inventory them, give them a GPS location, record data that can be utilized for their relocation process, which is what we did in the next phase. So you can see here, coral with an ID tag, a scale bar, and notice on the scale bar that there, it doesn't show up well, but there's color around the edges of it, so that you can then use computer software to then analyze if the coral, if the zooxanthellae density has gotten greater or decreased, if the coral is healthy, paling, bleaching. It's just one of the other tools in our box of things that we can do to assess the health and well-being of a coral over time.
Joe, can you go back to that? Okay, so when you identified that coral and you were relocating it, you didn't take that at the base and relocated it, you fragmented that one?
Okay, so the question is, did we take part of this coral colony or the entire thing? The answer is we took the whole thing. So, and actually under that hollow edge on the front side, really big chisel, really good hammer, and a lot of will. But the idea was that we wanted to move whole colonies at first, because we wanted to keep them intact and so that they maintain their natural size, and ideally they would then spawn within this year as well. So that was the idea, because if we fragment them, there's a relationship to the size of the colony and the number of polyps along with age as to whether a coral will spawn or not. So by leaving them larger and more intact, the likelihood of them to spawn is significantly higher. There are corals that we did fragment and then they'd turn into microfragments as well.
How's that one doing?
I'll let you know in December when I come back for monitoring. Probably pretty good. I can't tell what number that one is. Actually, 143 is doing great. I took a picture on 143 today, so that one's doing well. And this is at the donor site. This is then being transported and tagged in just an underwater gear crate. That was the best way to go about moving them, because we're extracting these by hand so that we can then make sure that we're minimizing damage to the actual living coral tissue. And then same thing, we try to handle them as gently as possible. A lot of times when these projects are done, they get a big underwater, like a big crate, and then just heap everything in the crate together. They're all hundreds of thousands of pounds of coral all grinding on each other of different species. Probably not great. So we tried to keep them individual or at least spaced out so that they're not bumping into each other, that they're not fighting each other the entire time. But you can also see that we transported them with an ID tag so that we can keep track of every single coral as we relocate them throughout the process. Here's actually some of the team from Saba with the lift bags and the corals, helping transit them from the seafloor to the surface. We looked at several different aspects and several different ways to actually try to get the corals from the seafloor to the surface. In the very beginning, we looked at trying to make like a coral elevator. But we realized that if we want to ensure the health of the coral and make sure that they don't get flipped over when they hit the surface or fall out of the bottom of the basket, that we need to treat them with the utmost care. So it was a little bit riskier for us as the dive team to do it that way, but in the end, it's probably still best for the coral health and their long-term survival. So there's 143, post-outplant at the recipient site. And it's charging right now, but I took a picture of that one today. So questions about how we picked which coral. So we looked for a set of factors based off of the health and then their relocation risk scores. And so we essentially we were asked to come up with a way to justify which coral should get relocated versus another. And so we said, all right, that's probably pretty important that we understand the why and the how of who we pick. So we focused on picking corals that were ideally exceptionally healthy, no signs of disease, minimal predation, a large amount of tissue to harvest, and also ones that would be extractable with minimizing risk of damage to the coral tissue. Also, the size of the total coral colony versus the amount of living tissue. There were several colonies out there that are, the size of a small car, but the only part of it alive is about this size. And the amount of effort necessary to extract that part versus just go find a healthy colony of the same size, we erred in the side of caution, and as opposed to trying to spend hours chiseling away from what has probably died back and is a less healthy coral, we identified and targeted the healthiest corals we could, with a handful of exceptions, one of them being this. This is one of the few pieces of Dendrogyra I've seen alive in a long time, and it's sad that that's all that there is, but it's great that we were able to find one, because that was actually missed on the initial environmental impact assessment that was handed to us before we started our survey work. So we identified this one and then extracted it, and it now is living at or at least was living at the coral nursery. But you can see that the difference in the health from November, because we had a late bleaching in the northern part of the Caribbean last year, well, in southern Caribbean as well. So we intentionally let corals recover till later in the year before relocating them, because we did not want to move them in their most stressed state. Another example would be this guy. And so you can see in the left photo from November that it was extremely unhealthy. The one on the right-hand side, the text got eaten, but I think that's March of this year. So it took about 4 months for it to completely recover to a state that it was then deemed healthy or safe enough to be able to relocate. This is a breakdown of the size breakdown of how the corals that were relocated, and so we tried to make sure that we got an even spread of all the different bin sizes, because what that means is that the different size classes are also different age classes. So we wanted to make sure that there are newer corals that have settled, grown out in the past 10, 15 years. We want to make sure that there's corals that are 50 to 100 years old, and then we tried to make sure we got a handful of really big ones, because if they've been around that long and they're still doing well, then we should keep those going. And the team had picked up and did even more large corals since we left. So the amount of effort that's gone into trying to preserve the biodiversity from this area has been exceptional. And then another one, this one's also doing really well. I took pictures of this one today. And so you can see when it was first inventoried, extracted, planted, and then monitored again, and it's in that bottom photo from April 11th, it's already growing over the fixative. At this point, you can't really see the fixative. That whole margin has now fused with the base rock, and it is now one with the system, which is exactly what we want to see. This is just a distribution of the corals where they ended up by species. So you can see there's a large number. We tried to get an even split of both Orbicellas and the Palmatas to both the recipient plots, also with a portion going into the nursery to be banked for future propagation and continued restoration on Saba. So here's just some examples of corals that were then collected to be stocked in the nursery. So here's the donor colony before any pieces have been extracted or propagated. We would ideally select only a small portion, ideally about 30 centimeters or about a foot long, that would then allow us to then produce enough ramets or individual fragments of that one genet to then be able to propagate it over time without having to go back and get another piece from the original donor colony. And that you can see here, which I think that's my hand actually. So that is a donor piece that's going to then be tagged and stocked in the nursery. And so that one will then be propagated over time to then create more pieces. And then as those grow, they just can continually be propagated. But again, back to the point that was brought up earlier, these are still clones of one genetic lineage. So it doesn't get us new genotypes yet. But if these all continue to survive, thrive, and then reproduce, they will then produce new genotypes and new genetic individuals of coral that will then, some are not going to make it, and then some will be the survivors into the future. Again, here's just part of the extraction process. The next slide's even better. It's a little messy. I think that's me and I think Bob extracting those pieces of palmata. But then you can see the fragments after they've been generated growing in the nursery. And you can see again this growth margin where it's that white. That is all new skeleton and tissue being laid down. So the middle part is actually the dead part of the skeleton where we bisected it and cut through it with the saw. And so you can see the coral is now wrapping over that skeleton and then reclaiming everything with living tissue. And so this is the difference in 2 months from partly healed to completely grown back over. This is just the species distribution of corals that we took to the nursery. The next picture is better though. So these are orbicellas. You can see a little bit of everything. So there's whole colonies, there's pieces of colonies, there's microfragments on fusion structures, there's donor brood stocks on plates for future propagation. On that one table there, it's not quite enough genotypes, but that's a subset of almost everything you'd need to put back most of a native Saban reef. And that was the idea, was to produce enough broodstock and enough also parts of individuals to then be propagated into the future and then keep this kind of process going. Here you can see the team working then on the nursery tree, then stocking all the new corals into the nursery. Again, doing very well. I think they cleaned them today, actually. We went by and there's some that are doing not great, but there's some that are doing phenomenal. So, and that's a difference in some within individuals, in different genetic individuals, but even within a single genotype, some pieces will do better than others over time. That's why we always try to make sure that there's a minimum of 10. That way that there's then enough pieces to then continue to propagate over time. You have a question back there.
You have to clean
The trees. The corals from the... No, you don't want to scrub the coral, but the thing that you might want to scrub would be this little margin here where there's like algae growing. But if it's happy and healthy, it will produce compounds to abrade or essentially chemical warfare away the algae, and it will start taking over that territory. But the main thing is actually cleaning the trees and the lines so that the fouling growth doesn't start trying to compete with the coral, and also so it doesn't start aggregating things that would be problematic like fireworms or coralophilia, coral-eating snails. Very good question. There was, did I see another one or no? Okay. Very good. Moving on. Again, just nursery tree stocked with donor fragments. These are then pieces to be propagated and essentially just as a genetic bank so that there's just an extra piece of everything in case something happens, that there's always a spare part. And then this is our post-relocation ecological assessment. So this is part of again, pre-survey, and then we do a post-survey as well to see what has changed from the beginning to after. So you guys have seen a little bit of this image again, but that coral right there is orange 18. That's the one from the previous images in the photo mosaic. That's it now at the recipient site, also doing well. I took pictures of them today as well. So as of June, there had been only a handful of corals dislodged, but no mortality at that point. Today I did see a couple that had not made it, but that's to be expected. Our target survival for this project, we're aiming for 80% or higher. The normal accepted rate for these kinds of projects is about 20 to 30%. So we're hoping to beat that by leaps and bounds, and considering it's been hot already, and the vast majority of them are still doing well, when we come back in December to do our long-term monitoring and we'll actually account for every coral, we'll then have a way better figure. But at this point, if I had to guess, it's still at least 80, if not probably 90% survival out of the corals that have been relocated. So there are some that are stressed, there are some that are bleached, but there's also the corals that were already there that are also stressed and also bleaching. So it's not like the relocated corals are underperforming at a higher rate than the native reef. Another one of the really cool ecological effects is your total fish. And so what we saw is that from before and after, as we relocated corals from the Black Rock Harbor site to the recipient sites, we saw a change in the total amount of fish and fish biomass. So already responding, which is pretty interesting. And the statistic that's missed out of here is also the types. We do break them down by clade, and it gets to be really boring and statisticky, but the important part is that, and those that went diving today saw it, a lot of neon gobies, which are a cleaner fish, and set up cleaning stations. Because the time window we relocated the corals, those fish were then recruiting throughout the spring. And they preferentially recruit to orbicellas and brain corals, which we moved a lot of. So there's now dozens of new cleaning stations at this site at Hole in the Corner where there were not before. And you can just see it in the fish behavior, that there's barracudas this size just parking over a coral, opening their mouths so the little fish can go in and clean. So it's really cool. And that, for me, that is a almost intangible effect of ecological success, that your ecosystem is building back those positive feedback loops that are necessary to maintain the health over time. And again, this is total coral. This one, we kind of expected this, that as we surveyed before, the blue is again before, the orange is then after. The diversity and the total coral coverage in the donor site went down, and the coral coverage in the recipient site definitely went up. And that's just from the sheer number of coral that we relocated before and after. Again, a little bit of a breakdown by size. So again, we increased the number of corals of each size class in each plot at the recipient site, which in the long term will hopefully lead to more resilience, more spawning, more reproduction, and then more larval production to then naturally replenish what's been lost in the reef. And then here's just the change in coral composition from the donor site. So you notice the left-hand pie graph has got more colors and more slices. The right-hand pie graph has less. And that's just the data visualization of the number and the species that we extracted from the donor site and relocated to the recipient site. But the next one's even better, because this is the change in composition at the recipient site. So you can see that one plot was almost all Porites asteroides, which is the mustard hill coral. It grows. Some of them can grow big, but it's not the most critical to what we consider reef building and needed ecological success. And then you can see the increase in species diversity on the right-hand slide. So the reason that that's really important is that is a big player for resiliency. Again, with if you have a monoculture, if your one lineage goes down, you lose them all. If you have 3 species and 2 goes down, you only have 1. If you have 10 species and 2 go down, you have a significantly larger cushion. And so that was another one of the goals, was to transfer not just the individual corals and species, but also to make sure that that biodiversity also got translated into the recipient site from the donor site. Same thing with the other plot, and you can see again, species composition changes from before and after, just as a result of all of the corals that have been extracted and relocated. Yes?
I note that the first species listed in the baseline, the APOL, the
Yeah.
site, and 1W or 1E are not located in the poster location at all.
Yeah, and that would be because we did the surveys as transects, so we laid out a tape. We didn't plant one where we laid out the tape. We probably should have, but that's also cheating. So we try not to do that, and that's one of the reasons that we try to make sure that All of our surveys stay unbiased. So when we lay out the tape, we do not cherry pick the best sites or the best line, if you will. We try to get an even assemblage or an even slice of what is actually there. Because if you only go over only the healthiest parts, then you create this artificial rubric by which you then build everything else incorrectly. So that's the reason that we moved a bunch of them, but you didn't see them in the before and after graph, is because there just wasn't one in the transect line. And then this is just a little friend. You can just see his mouth there. It's a little golden tail moray that from the Relocation. And I think that is my last slide. Thank you. So at this point, open to all follow-up questions. I figure this part will be more Saba specific, but I'm happy to answer questions until y'all get tired of listening to me. Okay, over there, and then I'll come to you.
With your coral trees, it would seem like it's a buffet for coral predators. How do you protect them?
Very good question. So the question was, the nurseries seem like a buffet, which they kind of are, and how do we prevent that from becoming problematic? So part of that is the cleaning and making sure that the structure itself does not grow a lot of large algae and fouling that then allows other organisms like the snails and fireworms to recruit. There's also the reason that most of them are either suspended off the bottom, either off PVC tables, or in this case with a rope, because it then just becomes significantly harder for the predators to then get to them. Fortunately, in the Caribbean, we have species of parrotfish that do chew on coral, but they are not dietarily restricted to eating only coral like you would run into in like the Indo-Pacific, Australia, where there are obligate corallivores, things that can eat only coral. So for the most part, by just taking them away, one of the things that isn't in a slide that's part of our rubric for site selection is making sure that there are predators of the predators. So things that eat corals like fireworms and corallophilia, we want to utilize sites where there are triggerfish, sand tilefish, white grunts, white margates, and what was the other one that's on there? Hogfish, that will eat all of these invertebrate predators of the corals. Very good question, though. Let me go back to the last slide. That was pretty. Okay, yes.
So can we kind of think of this, if you took 1,000, roughly, corals from the donor site, you moved them to hole in the corner, can you give us some kind of estimate of how many of those are now on the substrate, secured in with your fixate, whatever you call it? I'm thinking storms, so lived here too long, right? Mm-hmm. So if we get a storm next week, hopefully not, those that are there should be secure. What percentage is on the trees? And do you have a mechanism of preparation?
That is like a lot of question for me to repeat back. Oh, okay, so the question was, what percentage of the corals are affixed to the seafloor versus what percentage are not permanently affixed in the nursery? The easiest answer to start with is that everything that's in the nursery has already been planted. So the piece that's in the nursery is just a donor piece. So the original colony has already been planted. And so out of the 1,000 plus are on the seafloor. Everything that's in the nursery is just a piece or a subset of an already planted coral that can then be used for future propagation.
Lost the trees,
they're still on the reef.
Still pretty protected by what's fixing it.
Exactly. And I originally put into the proposal to have 3 baskets to put every coral into, so that there would be 2 different nursery sites, but it was time and logistically prohibitive. And in reality, knowing that each coral has, there's a copy of each coral that's in the nursery, there's a piece of it on the reef. So if we know where they all are. So if we need to go find, Orbicella blue 143, we know where it is. so we can go back and get it. So we could potentially lose everything that would be in the nurseries, and other than the cervicornis, which is separate from our part of the project, and we could still go get every single one of them again. Okay. Okay, we start there and I'll work my way.
I'm just curious, how many corals are there in the original plot?
Okay, that's a great question. How many corals were originally in the donor site? To be frank, I don't exactly know. I could guess, but it would be, we've moved 1,000, and I would guess that there are more than that. But it also depends on the species and size. If you're talking about recruits, there's probably 5,000 recruits smaller than that. Now, number of coral colonies bigger than this, we've probably moved, I would guess, at least 30% to half of the potential donor corals. There are a lot of corals that are there that are in decline. And there are actually many corals that we inventoried in the beginning of the project in November that then died off by the time we actually got to the relocation phase. And new corals were then added to the inventory from the healthy stock there. But that was a great question, is what percentage of the corals were relocated from the donor site to the recipient site? Yes.
The site that you remove the corals from, naturally marine ecology is gonna go down, correct?
Correct.
Because there's nothing there. So what's gonna happen there?
So the question is, so what, now that we've moved all the corals, and then today we moved 68 conch from the donor site to the recipient site, so they can continue grazing. So everything, the donor site is going to become a harbor.
I understand the development is good for countries. When you put a harbor there, are these people gonna do something continuously to help the ecology at the harbor? Or it's just gonna go, you know?
That is a great question that is above my pay grade. So as far as Coral Vita is concerned, we were
I wasn't sure if they, because companies like resorts, they let you come in, they move the mangroves and corals and whatever. However, where they remove it from, they have systems in place to help to replenish it. You know, they just don't just remove it and turn it into a beach. There's systems.
Okay.
So I'm just,
So the what's going to happen long term is that all of the corals that are then in the nursery will then be propagated, replicated, and then outplanted as part of Saba's continuing coral conservation and restoration program. As for what the contractor for building the harbor is dedicated to doing after that, I don't know. But
how far are both sides? Where you removed it from to where you're putting it?
about 1.8 kilometers plus or minus. And so let me go back to that. Let me go back to the map. Backwards. Let me see if I've got the big. I don't know if I've actually got a whole Saba map on here. I don't. But here we go. So one of the reasons as well that this particular site was chosen is because Saba in particular is, you know, a volcano, and it's tall mountain abutting right to the shoreline. So the sun angle on different parts of the island is completely different. So the sun angle from, you know, out at Torrance Point is different than it is at Tent Reef, than it is from Spring Bay. And so because we wanted to try to weight this for the best success possible, the decision was made that as opposed to trying to move them to a different side of the island, trying to pick the healthiest site within that matches the same sun arc, that they get more or less the same amount of daylight hours, more or less the same prevailing currents, more or less the same conditions. Whereas if they were to go over to like the Ladder Bay side, the ecology of Ladder Bay is completely different. So there was a question in the back.
How long were you here for altogether? Like from the time that you were contracted to do what you've done and how long? Like 2 years?
Between some and enough. The question was, how long was I here? So our leg of the project ran from October of 2024, which was development and planning, surveying in November and December of 2024. Relocation started in February of 2025. And our hard stop date that we were given was the 30th of May. We managed to complete the initial 250 coral contractual relocation, I think around the 21st or 22nd of April. And then we continued relocating corals into June as we were also doing our post-relocation surveys.
Are you obligated to keep tracking the progress and the health of the coral for a certain amount of time?
The question is, are we mandated to continue monitoring over time? For a given period of time, the initial contract stipulated at least 18 months of monitoring. That's what we're required to do. We're going to keep coming back and monitoring Saba until my legs no longer work or Coral Vita doesn't have money to send someone. Because this is information that we can always use to develop and get better about our processes and ensure that the success, really, to be able to realistically ensure and assess what our projected success rate for these kinds of projects would be.
Which kind of follows up to my other question being, are you happy with what you've achieved? Is this anything that you could have done better? And have you learned anything that you can take on future projects?
That is a lot of questions in one question yet again. Okay, so do I think we could have done it better? Yes, I'm the more perfectionist you can try and be, the better, but perfection is also the enemy of progress. I'm very pleased with the results that we achieved, and especially considering the lift timeline and constraints and how difficult it is to do these kinds of works on Saba. We couldn't do relocation operations in any day when the wave height was more than like 0.8 meters, which is most of the time on Saba. So given the constraint of calm and flat days, I would say around the middle of March, my anxiety was at an all-time high as the person responsible for executing this project. And by the time we got to April and had moved 250 corals, I said, excellent, we're doing more. And fortunately, the team has continued to do more since our contractual obligation ended, and we've left this part of the project in their very capable hands. So the real answer is yes, there's several things retrospectively that I think we could have done better, but one of the biggest limitations was the actual stipulation of generating an inventory before we were allowed to start relocating the corals, because that made it significant. If it wasn't for our mapping tool, it would have been impossible to go back and find every single coral. And we inventoried initially 450 corals, and we moved 387. So there are corals that were inventoried that were either not relocated because of health reasons, or we didn't find it again. Which is just a part of the process. And that's one of the reasons that we're developing the underwater tech, so that our app will then actually have a GPS transponder so that you will then be able to see the map underwater. But that's something that, the time, energy, and tech would have cost 10 times more than the funding for the project. So if it's do everything perfect or do the best for the ecosystem possible, I feel very good that we did a good job for the ecosystem as possible. Hang on, there's two more pieces. What was the second?
Is there anything that you've learned that you can take on for future projects?
Oh, absolutely. There are so many things that we learned in the process of doing this project that will transfer not only to our enterprises, but again, after we continue doing long-term monitoring, we're hoping to start turning, all of our procedures into a set of guidelines that can be utilized by other practitioners. Because again, the survival rate for most of these projects is around 20 to 30%. And the reason for that, and I'm probably gonna get in trouble for saying this live, is that usually the marine biologist leaves with the EIA, with the environmental impact assessment. So there's an assessment made beforehand. Someone circles a spot on the map and goes, they wanna build a thing here. There's a bunch of coral, they should go there, and then a team of commercial divers, local divers, whoever it may be, is then contracted by a subcontractor to then actually move the corals. We actually had a great example of this in Grand Bahama, that Carnival was building a new cruise port. We applied for the contract. They hired another company. Their success rate has been not good, and to the point that some of their divers came to some of my staff and said, hey, we're pretty sure we're doing this wrong. Can you just tell us anything to make this work better? So for us, we're actually trying to develop a series of guidelines and rubrics that we can use not only for, again, for our operations, but also bring up the standard of what it means to do these kinds of projects. Was that everything?
Yeah,
Am I good?
sorry, I'm going to stop here. Thank you.
Okay. Yes.
So post your contract, you said they've moved a lot of additional corals. So is that all tagged and marked into your system so that you can go back and monitor the health of that as well, or not?
No, that is because they are not, they have a user-facing copy that we generated as part of our final report for the project. But our software is, what's the best way to put it? Expansive. It's more than most entities currently want to pay. We're also working on trying to parse it down and turn, not everybody needs our full inventory suite of capabilities and task generating for land-based coral farm related activities. So we're developing actually a parsed out version that is being put in play by Reef Renewal Bonaire now, that utilizes just our nursery inventory function of the app. But the degree of difficulty, I mean, for me, if I had to pick one of being able to keep, the laser focus on each individual coral and where they are versus moving coral in bulk, my natural instinct is to move coral in bulk, not necessarily, try to keep track of who's who for the important ones, but everything that can go must go. But our requirements for this project were slightly different. So it's good that we were able to target the bigger and the more critical species to then leave a lot of cushion for the team from the SCF to move everything else. And so the upside is that we did our post-photo mosaics the week after we finished moving corals. So when I come back in December to do photo mosaics again, we'll then capture all of those new corals that have been relocated since our post-relocation, and we can at least then track those, give them an identifier within the map over time, and then keep track of them over time. Good question. We start there?
Yeah, I mean, really impressive work, Joe, and I especially appreciate your commitment to continue sampling through time and post-monitoring. I'm just curious, one of your goals is to save those colonies that would have otherwise been destroyed. Another is to perhaps increase coral cover and diversity in the recipient site. Do you have any control plots within the recipient site to let you see how those will differentiate through time?
Yes, and unfortunately, it's not on, the transects are not on the map, but so you can see there's the red box, which is 3W. The purple box is 1E. There's a 2C in the middle there that has transect 2. That is essentially our control plot. Now, at this point, there are probably some corals that have gotten relocated into the control plot, but at least for our initial before and after, we did have that to weigh against. So, and that's what I used when I was looking at today, like how many of the corals that we relocated that are tagged versus ones that are native versus ones that the SCF team has relocated. And it's pretty easy to tell because the fresh ones still have the fixative baseline. They still haven't accrued over all of the fixative yet. In about 2 years, it's going to be really hard, but that's the best kind of problem to have. Yes?
What's the legal requirements of making a coral garden?
What are the legal requirements of
Or
making
legal,
a coral?
Like, are there rules or regulations, laws, or?
So the question, are there laws and regulations governing?
Do I need permits, or do you know this?
Here, specifically here, because we
Yeah,
All right.
we started working here, and my plan is to make a coral garden. Like, is this legal to do, or?
All right, so let me work backwards. So the question is, can I just start up a coral nursery or coral garden? Again, that's one of the, I'm going to take the cop-out answer and say it depends, because it depends on where you are, the regulatory framework. In the US, the answer is a hard no, unless you're a research institution, a coral researcher, or an entity that has the money, power, sway to get that level of permitting, usually through NOAA or National Marine Fisheries. It's quite a series of hoops to jump through. If you were in nowhere, Micronesia that no one cares, no one's probably going to tell you no or stop you. It's probably in the books that you're not allowed to, because most corals are either fall under CITES or an endangered species handling. So you at least need a permit to handle them, much less do anything else. But it depends. Here you would probably, I would guess you'd have to go through a combination of public entity Saba, the Conservation Foundation, and then I would guess that Marine Fisheries in Holland actually has there's someone else that has a bigger call. There's several different parties, and I'm not actually sure whose call that is here, but most places there's multiple loops that you have to get connected, or you just have to be somewhere that no one really cares.
Can you clarify that's because you can do more harm than good?
Yes, okay, and so the reason for that, it sounds a little bit elitist and kind of like a dick move, and the reason for that, the reason that anyone, anywhere is not allowed or usually not even encouraged to just go start doing coral restoration, gardening, those kinds of operations on their own, is that there is a big risk. There's a lot, even for us quote unquote trained professionals, there's still a degree of risk. But one of the biggest problems is that without any regulation or oversight, and Indonesia is a great example of this, that you will have overlapping projects that are actually detrimental to one another. So someone will go out and set up coral tables, and then, or set up like the rebar domes with the corals on them, and then another organization will come through and just start dropping blocks of artificial reef. And because there's not enough oversight, knowledge, or introspection into making sure that you're doing the right thing, there have been several cases in the last 10 years or so of exploitation by operators or entities that kind of don't know what they're doing, that end up actually extracting and having more coral die than they actually propagate. So they end up being a net negative as opposed to a net positive.
I did in the past, a fish break coral off, and like you always see small pieces of coral just laying in the sand. Like, out of experience, these corals will not survive. So breaking of corals, of course, you should not really do that. But if along the dive, as a dive guide, you know, if you find small pieces of coral just broken off by parrotfish or yeah, then you can.
That's what we did in the morning.
Exactly, you can pick this up and just put them on a small rope or like on a structure.
But that's very dependent. Your ability to do those kinds of things, to take what would be called fragments of opportunity or naturally produced fragments, is still dependent on where you are. If you're in Hawaii, no.
It's based on this, but
Yeah.
In collaboration with the SCF, there are
Okay.
probably possibilities.
I will give a knock and,
Yeah.
and see.
Those ideas and efforts,
Yeah.
I think, are definitely appreciated.
I'll come by.
But of course, in collaboration.
Cool.
Anyone else? Oh, fantastic. Oh.
Big round of applause for Joe and for the efforts of the Saba Conservation Foundation. Thank you everyone for coming. We invite you to stay for dinner at Liam's Cuisine, and our next presentation is this Sunday, October 5th at Calibri. Enjoy your weekend.